Welcome everyone to another Dolphin Communication Project Deep Dive webinar. Today we are joined by Joëlle De Weerdt, all the way from Belgium. And she is a PhD student in marine biology there, but she does her research off the coast of Nicaragua. She is the founder of the Association ELI-S. And while her main focus is on humpback whales, today, she is going to share with us some of her work with bottlenose dolphins and how photo identification can tell us all sorts of things and not just be a pretty picture like we have there. So if you are listening live, please go ahead and use the chat function for your questions. You will find yourself muted. That is on purpose. And with that, I will pass it over to you, Joëlle.
Hi, Kale. Thank you so much for the invitation. I'm very happy to be here and share about my research. Can you see my screen?
Yes.
Yes. Okay. So yeah, you said it all. I'm doing my research in Nicaragua, but I will dive a little bit about who I am, because I think it's nice to talk about the dolphins, but it's also nice to talk about the person to understand how I got there, you know. So I'm a person with a childhood dream. I think like most of us, to protect whales and dolphins. That was my main driver in my research. And it all kind of started in 2010 when I had my my master in biodiversity and ecosystems in Belgium. And then I've been volunteering in many different projects on whales, but also about dolphins in Slovenia, Great Britain, in Mexico, and in Madagascar. And after I've experienced many things in research, but also in education, I decided to create my own project. So I learned quite a lot, and I really wanted to condense everything in one project. So the project is scientific research. So this is a cetacean conservation project of Nicaragua, and is composed of 3 different programs, because I do believe that science is important. That's why I wrote in big, science for a purpose. I really want to do research for a purpose, because many times I went to conferences and I see people saying it's important because we need to save the animals and stuff, but then it needs to go out to the general public. And therefore, I combined science with education. So I have an educational program in Nicaragua. So the project that I developed is in Nicaragua, in Central America. And I created that with my own nonprofit organization called Association ELI-S, like you said. And ELI stands for Education, Liberty, Independence in Science. The project is specifically in Nicaragua. Why? Because I knew there were humpback whales migrating there, and there was little information available on cetaceans in general, and that's why I created that project there. And so the education program consists also of educational classes with kids. I give information sessions to fishermen. So I'm really involving quite a lot the local community. So therefore, in 2017, I received an award from the Yves Rocher Foundation, which is a company that created a foundation, but it's a company that makes makeup based on a vegetal basis, so they're not using animals. And they gave me an award because I was doing research and involving local communities. In 2018, I got another award for just the similar reasons, because of this research and involving local communities by Ecology Project International, sorry. Then in December 2019, but I said 2020, I started a self-funded PhD project. So with all the data I've been collecting in Nicaragua, I just decided to start this PhD because remember, it all started with a passion for science, and that's still the core of the 3 programs. I forgot to say the third one is the conservation program. So of course, it's science education for proper conservation of whales and dolphins in Nicaragua. In the conservation program, it includes like beach cleanups. It includes also the fact that I give an annual report to the government about what I'm doing in Nicaragua. In 2021, I received from the Jane Goodall Institute the Young Researcher Award. That is because I am studying the impact also of anthropogenic activities on humpback whales by assessing also the whale watching rules in Nicaragua. And in 2021, I forgot to write that, I also helped the government to establish whale watching rules in Nicaragua for the first time. So that's a pretty big advancement in those 6 years. And then in 2022, I started collaborating with bigger organizations like WWF, IUCN, and IWC, the IWC's International Whaling Commission, where I was invited as a national expert to talk about whales specifically and also other cetaceans. Oops, I'm sorry. 2023, this year, I was invited by Whale Tales. That's an event in Hawaii to talk about humpback whales in Nicaragua and Central America. And then I received an award for a brilliant marine research idea. That's it about myself. So just that you know why I'm making this presentation today, and that's it. Okay, so just to make sure that we all know where Nicaragua is located, I added this little video where you can see Hawaii, the United States, Mexico, and we're zooming into to Central America and specifically Nicaragua. So here is Central America. Central America is composed of different countries along the Pacific coast. I'm not going to talk about the other side of Central America. So you have Guatemala, you have El Salvador, then you have Honduras with a very, very small coastline. We have Nicaragua, that's located really in the center of Central America, and you can see it's a very big country with two coasts. You have the Pacific and the Caribbean. You have Costa Rica, that's a bit more famous than Nicaragua, a little bit more. And then we have Panama, which is mainly known for its interoceanic canal. Sorry. So the study area, now we know where is Nicaragua. When I started the project, I wanted to know what was going to be the impact of the potential canal that they wanted to construct between the Pacific Ocean and the Caribbean Sea. Now it didn't happen. It was supposed to start in 2016 and it just didn't happen. But at that time, I didn't know if it would happen or not. So I designed the project in 2 different study sites. So the northern study site is Padre Ramos with the red arrow. And that was my control site to say, like, there would be no impact of the canal, supposedly. It's a natural reserve. Weirdly, there are people living in this natural reserve. So it's not like we would imagine that it's preserved, right? So there's people living there. They're still using illegally some bombs, and there is only national tourism in that area. So it's quite undeveloped in the area. And then we have San Juan del Sur, where I focused my research. As you can see in the northern part, I was there only 3 years. And then I've mainly focused in the southern part. So that would be the impacted side by the canal. It's a very touristic place. So there you have national and international tourism. And in the fishing activity, there is no use of bombs. Fortunately, that's really good. So this is San Juan del Sur, like you can see on the left side. I put also a little picture of the boat we're using. It's a fishing boat. They call it a panga. You can see me standing there with my camera. That day we were looking after bottlenose dolphins, and well, in this case, I put also a picture of a humpback whale. It's going to be the only humpback whale, I think, from the whole presentation. So, but from now on, I will focus on dolphins. So what is a dolphin? I was just thinking, like, I'm going to talk about dolphins, and you know, I'm always so much focused on whales that I was like, maybe I should replace everything again. So cetaceans are composed of mysticetes and odontocetes, so 2 big groups. The mysticetes, they have baleen, so that's that structure you see on the left side. In their general behavior, they're solitary. Of course, they can group in function of the if it's a reproduction or if they are feeding, but they're not sticking together in groups, and they eat krill or small fish that they filter through the baleen. And then you have the odontocetes, so they clearly have teeth. They're living in groups, and they eat a medium to large fish generally. So the ecology of both groups is overall pretty different. And interestingly, well, I wrote this also because I was thinking of dolphins. What is a dolphin? What is a whale? Because if we say a killer whale, people say it's a whale. While I would say killer whale is a dolphin because it's an odontocete. So it's a bit the scientific perspective on how you classify things, and then the popular way of classifying things. So that's why I was just introducing very briefly this classification. So the most common dolphin species that we see in Central America in general, well, also in Nicaragua, are the spotted dolphins. So like you can see on this left picture, you can see that clearly that the dolphins have spots on their back. So that's why they're called spotted dolphins. Their Latin name is Stenella attenuata. And then you have on the right side bottlenose dolphins that probably most of you know the best. And we are collecting data on both species. We have occasionally also false killer whales. There was an opportunistic sighting of a fisherman about killer whales, but they are pretty rare in general. So those are the two common species. Oh, I just said it already. So the spotted dolphins, they have spots on their back, as I said. They're small in size, and they also have a longer beak. And then the bottlenose dolphins, well, they have no spots on their back. They're a bit bigger. Now you need to have seen the dolphins quite a few times to understand what I mean with bigger. They're not like huge, but slightly bigger, and they have a shorter beak, like you can see on this picture. Now, dolphin morphology, well, I actually introduced it already in the previous slide, so that we understand the following slides. So first we have, well, the beak, then we have the blowhole, they have eyes, well, there you have the pectoral fin, then we have the dorsal fin, and then the caudal fin. So the dorsal fin is going to be the element that we are mainly using to identify animals. This animal is called Pepito, by the way. It has a little name. One of our favorite dolphins. So one of the main research methods used by marine mammal scientists is photo identification. And it's a great tool because it's non-invasive, and you can use the natural marks that are on the animal. So you go on a boat, you have your camera, you find dolphins, you make a picture, and you can build up a database and catalogs. So with that, you can do a bunch of things. And many researchers, well, all over the world, in Australia, in the US, in Slovenia, wherever you want to go, where there are dolphins, they have these catalogs. Now, I put a fingerprint. It's actually normally something I use for humpback whales because their tails are unique as our fingerprint. For dolphins, it's not that. I'm a bit allergic to say fingerprint because their dorsal fins are changing from the moment of birth until the end of their life. It's stable, but it can change. While humpback whales, they have this pattern. You come back 20 years later, it's still the same whale. So it's not as perfect as a fingerprint, but it still helps a lot for identification, and scientists can do a bunch of things with that. What we're looking at are the nicks and the notches on the dorsal fin. So if we look at Pepito, you can see that there are like some parts of the dorsal fin that are missing, and that makes it unique, and that's why I know it's Pepito. All right. What can we learn from pictures? Well, basic stuff. I promise there was no humpback whale anymore, but I see there is still one. It's gonna be the last one. Well, with pictures, you can do species identifications. That's like the first thing. On the left side, you see false killer whales. In the middle, the bottlenose dolphins, our famous Pepito. And then on the right side, we have the humpback whale, right? So that's like the first start with pictures. And then with pictures, we can also define how big a population is. So when we go on the boat, we try to find dolphins. Well, this applies also, of course, to other cetacean species, not only dolphins. And basically, when you go on the boat, you do a sampling, right? And so because often when I do research, people ask me, hey, do you mark the animals? Like, do you label them? And we do not label them. Just with this picture, that's their own label. So usually when you go with a label, when you go in the field, for example, you can catch turtles, you put a label, those turtles are leaving, and the next time you come again and you have other turtles, maybe with a label, maybe without a label. And then in this way, you can calculate how big your population is. And that's exactly the same system with the dolphins, but it's a bit less easy than catching a turtle. You need to catch a picture of their fin. So that's logistically not always easy, because you can actually already see in the two first pictures that I placed in this presentation. On the left one, it's a pretty dark picture. On the middle one, it's a fairly well lighted picture. So you see some scars, and then you see more details. So depending on the quality of your picture, on the angle of your picture, because there you have very good angles. But on the bottom picture, you see that the dolphins, where you can have water in front of your dorsal fin. So it's it's a bit frustrating, because sometimes you show the best pictures, but in reality, it takes you a lot of tryouts with the dolphins. You hope that they're turning around really well to have this picture. So it's really important they are parallel to the boat, so you can do these population size estimates. But basically, you calculate with the marked one, the recaptured, and then you apply this the formula, so you know how many dolphins you have in the population. So that's pretty cool. I haven't done that yet in Nicaragua, and we're working on it to do that in the future. Then another element you can do is, determine individual preferences. So I saw that Emma was talking about that. So if you want to know more about it, you can look at her presentation in another webinar. But basically, the question is, okay, who likes to be with who? Like, which dolphin likes to spend some time with the other one? And you have a lot of very complex formulas to calculate how dolphins like each other. And basically, on the right side, you get then a network diagram, and then you can see with the arrows who is linked with who. So every square is an individual in this case. So this is an analysis we're actually doing with a colleague of mine on the social structure of bottlenose dolphins in Nicaragua. And you see that there are two clusters, so two main groups, and within each group you have certain individuals that like to stick together more than with the other group. However, you can always have some exchanges between the clusters, but basically, based on the number of times that they were seen together, they will form a tighter group and more distinct in between each other. So that's one of another application of pictures that we're making from animals. With pictures, you can also know where their movement patterns. So where are animals going? I gave another example of the research I've been doing. So there is, we are working on this paper, so it's getting published. So it's actually not really official, this figure, but well, it's accepted. It's going to be published soon. But basically, by sharing pictures of the dorsal fins of the false killer whales, we saw that false killer whales from Baja California, so on the northern part of the map, are more related to California in the US. So you have a closed group. So you see that there is no connection with the central part of Mexico or southern Mexico. While you see that there is quite a few exchanges between Mexico, Guatemala, and Nicaragua, and the northern part of Costa Rica. So that's, let's say you have a second group, and then southern Costa Rica and Panama is another group of false killer whales. It's not clear if there are other populations, like genetically they're different, but at least with pictures, we can see how animals are moving around. Interestingly, in Nicaragua with the bottlenose dolphins, I have only a few recaptures between both study sites. I have to update the comparison between the two catalogs because I keep on generating new information in the southern part of Nicaragua, so we can see if there is the movement, how far the dolphins can go. And I think like they are traveling quite a lot in search for prey. And well, with pictures, sometimes you can also see prey preferences, no? It can vary according to the locations. On the left side, that's a picture we made in Nicaragua. And then I Googled some pictures with fish, and I could see that at a quick look, I'm not going to dig into the species identification, but you can see that clearly that our fish, well, in Spanish is jurel, is really different from all the other species you can see on the right side of the slide. I even found an octopus, so that's pretty surprising. I'm not sure if it was playing with the octopus or actually ate it, but I think it's really fascinating to see how diverse the preys can be for bottlenose dolphins. But with that, we can also identify preys. And then with the pictures, we can also look at impact of humans, but also from animals. On the left side, on the left side, you can see the first picture. On the top, sorry, you can see that this dolphin was probably bitten by a shark. That was a female, by the way. It looked pretty fresh, so that was a recent occurrence that the dolphin was attacked by a shark. On the right top side, you see, well, plastic pollution. You can also see how dolphins can interact with garbage, sadly. On the left bottom, you can see also this horizontal line in the dorsal fin, which is definitely not created by another dolphin. This was highly likely due to an interaction with a fishing line. So that shows that there is some interaction with fisheries. And then on the right side, the right bottom part, we have, well, those vertical lines on the back of the dolphin, and that is highly likely also the propellers of a boat. So that's why there are some regulations concerning the distances to dolphins that needs to be respected, because otherwise you can literally harm the dolphin. There was one picture that was a little bit more difficult to watch, because you really see the tail that was completely cut in two. There is a whale that was seen like that too, and that's very, very sad to see. Probably they do not survive. I didn't do a follow-up of that picture, but in this case, where the dolphin with the propellers will probably survive, that's not a problem. It's relatively superficial, but still it can impact the survival of the dolphin. But anyway, from pictures, we can assess all these different things. And I wanted to share with you that all this information that we're collecting, that's population size, the social structure, the impact, movement patterns, all this can be used for regional protection actions. And I wanted to share that in Central America, there is like some things happening for the level of the legislation for the conservation of marine mammals. So for example, with the IUCN, which is the International Union for Conservation of Nature, we and I say we because we were different experts from all the countries, put together all our data to say, hey, like this orange area is really important for humpback whales. I have also proposed another area for whales and for dolphins because I have this data. But for example, in the northern part of the country, I proposed to the area to be also an IMA, an important marine mammal area. And it was like not really validated because I didn't have enough information. So this scientific information is very, very important because decision makers need that information to validate a protection area. So that is like an important marine mammal area according to the IUCN. Now, it doesn't mean that it's at the moment it is protected, but that's a suggestion for the area to be protected for the good sake of the species. With WWF, last year we talked about the corridors. So for great whales, it's also whales. I'm sorry, it's a lot of whales, but it's just to give you the impact of the research for the animals. And there we defined corridors, so like really important areas for different whale species. So it can help to have new opportunities and actions to protect migratory whales. And then specifically for Central America, for humpbacks, they are endangered. So we were developing a management plan for Central American whales that we proposed for the International Whaling Commission. That's just for the humpback whales. At the moment in Central America, dolphins along the Pacific coast are not receiving a lot of attention. And I hope that it will change in the future. But we need to do quite a lot of research still compared to other areas where really like, doing all the basic stuff, like knowing how many dolphins do we have, where do they occur? And, so we have to start everything from scratch. So at this point, it's very exciting because there's a lot of things to do at this moment. And I hope that in the next years, people will take, also will give importance also to the dolphins. And the whales is just starting also. It's not, there's a lot of initiatives, but it's still also, it's all just starting. Anyway, so if you want to follow up with the project, well, you can always send me an email if you are interested in helping in the project. You have my website. If you want, we also have merchandising, like we have whale bracelets. I have mine today. Rings. You can also join us on our marine mammal research training program that we run from August to September and January to April. And do not hesitate to follow us on the social media. And thank you again for listening to me, being present today. And I can't wait to hear your questions.
Thank you so much for that. I'm going to wrap up our little slides and then we will get to questions. Wake this up here. There were a couple of nice little plugs in there. Thank you, Joëlle, for past DCP webinars. Emma's chat, if you want to look at that. If you're interested in those interactions between individuals, that sociogram that was up. Nicole has a good talk on that. Those clusters, by the way, in your data, Joëlle, are so cool. Our data in Bimini, we don't have clear clusters like that, so that was really fun to see.
Nice.
All of our webinars are posted directly on our website. You can find them under the Knowledge Hub, or if you prefer YouTube, they are there as well. And if you watch all of our deep dives and our dolphin lessons, which are geared toward younger students and the young at heart, and you still need more content, make sure you check out our podcast, The Dolphin Pod, also directly on our website or wherever you get your podcasts. And then just like Joëlle's organization, DCP is also a nonprofit, and so we would not be doing our jobs if we didn't remind you how you can support our work. So you can become a member, you can donate, you can buy swag in our online shop, you can join our director Kathleen in the field, follow us on social media, share our posts, like Joëlle. I heard a lot of DCP philosophy in there as well, right? We're not just doing science for other scientists. The data are important, and science being sound is so important so that it can get published, so that managers and governments actually have the information to make good decisions and justify those decisions. But we also want to funnel all of that science into education to lead to conservation, right? We all know that we really just can't protect what we don't understand. So with that, before I keep rambling, we have some questions. This one is probably kind of an isolated, or no, two are kind of related. So I'll ask you those first. The illegal fishing bombs that you mentioned at the beginning as an issue that's going on in the northern region. Is that happening actually in dolphin and whale habitat, or is that kind of peripheral to where the dolphins are?
Well, I don't have the proof that they are using it when dolphins are around, so I can't really say like, well, they use it when it's in the vicinity of dolphins. So that's definitely something I'd like to investigate in the future. But I've seen something interesting because a project was done by John Kalambokidis with Cascadia Research like 15 years ago or something in Nicaragua. And so he did a few surveys and had some information. And interestingly, in those 15 years, it's the same area, right? You really see a shift offshore of the animals. And I don't know why. I don't know if it's because they were using the bombs in the coastal area, and then the animals adapted and went further out. I don't know if it's related to bombs, so it's a huge statement. It's just a thought, I'm not sure, but it's very interesting to see this shift more offshore, and to explain it with environmental parameters. I don't know how I could explain it. Maybe due to the bomb. So I don't really have a clear answer to give you to evaluate how much it impacts. But also in the northern part of the country, we have more strandings. So I have no idea if it's related to those bombs. There are more strandings. I don't know if it's because there's a current that's pushing more toward the coast. It could be also just because of other parameters rather than putting everything on the back of the bombs. But it's just like something in my mind, like, okay, it could be this or it could not be that. You have to be cautious about your conclusions if you don't have the information, because you can just point at someone and maybe it's not their fault. And that's very important as scientists, that we're taking care of not pointing at someone and without having the proof that they did it. But those are small observations I have that maybe are due to bombs. I don't know.
Right. And while we're talking about human impacts, someone asked if boat strikes are common, and is bycatch an issue?
Well, for humpback whales, we define that vessel strikes and fishing gear entanglement are the main issues for that population. I've had some humpbacks entangled. I haven't seen any dolphins entangled, but I've seen a fisherman doing a selfie with a dead bottlenose dolphin on his boat. I don't know the whole story. He put that on Facebook, so maybe it was trapped in the net and drawn. I don't know. I don't think he actively fished it or something. But yeah, so we have no data about that. And when there are strandings, we don't have autopsies, because often I know about a stranding way too late. So the animals are or in inaccessible areas, or they're already in a decomposition state that it's too advanced, so you can't really do an autopsy. So like recently a humpback whale stranded, and it's just speculation about what on the reasons why it's stranded. I don't know, visually there was nothing wrong. Maybe it's a vessel collision, but you need autopsy. So again, it's a vague answer because we just don't have the data. There are so many things to do. And autopsies, like having a stranding network would be something very useful to be able to answer this question more specifically. But I mainly see the dolphins with marks from the fishing line. So there is interaction with the fisheries, but if they die in the water, they sink in the water. So you don't know if you don't have that information. It's hard to tell. And unfortunately, that bottlenose dolphin, the picture was not good enough to see the dorsal fin, so I couldn't check it with the catalog.
Don't know who it is. Mm-hmm.
Yeah.
The bottlenose, are they considered the coastal ecotype or the oceanic ecotype? Your picture of, was it Pepito? I don't know if it was the editing, looked like, compared to a bottlenose in the Bahamas, that looked very oceanic to me, but I don't know what ecotypes you have in the area.
That's interesting, because that was a question I was asking myself too, which ecotype we have. Now, yes, the picture is a bit edited, so the animals is a bit darker. That's maybe what you mean, right, with the color.
Yeah.
But apart from that, I've seen a paper about like really the morphology also of the dorsal fin, like they're thicker if they're offshore than compared to coastal ecotype. I've had like a few dolphins with this really thick dorsal fins, but I'm not sure. I won't tell you. I'm not sure. I think I'll have the coastal ecotype, but you say that maybe I'm offshore ecotype, so maybe we should
and that's
discuss
why,
it.
like, that's a reminder to everyone listening, like, you may see a photograph, but because you don't know what went into the photograph, editing and whatnot to make marks appear more obvious or whatnot. So I wouldn't jump to any conclusion on your images, but it prompted in my mind to say, oh, what are they here in the Bahamas? We have mainly the coastal ecotype. They're on the smaller side of average. They're quite light gray. They are close to the coast. Their groups can be bigger, actually, but I don't know if that's valuable. And then when we occasionally see an oceanic or an offshore bottlenose, like, it is distinctly bigger and darker and has like a pigment difference in their peduncle. So we do occasionally see them, but we haven't done any, extensive studies on them.
Yeah, so the area is a bit special because we have an upwelling system between January and April. So there is the Costa Rica Dome, which is nutrient-rich waters that is pretty dynamic spatially and temporally. So this dome is moving toward the coast and going offshore again. And I don't know if those animals are following this current. So potentially it could be an offshore ecotype and they come to the coastal area because they're following the dome kind of thing, or they're just coastal and it's just, a coincidence that they're around. However, I have data also in the rainy season when this dome is not there anymore. I have much less dolphins, but apparently there are differences in the structure also between the two seasons. So I don't know if there is a part of those dolphins that are coastal and another part that comes. So there's kind of a mix of both. That's what I think. Some of them, are. That's why. But I think I can maybe identify that with the residency time or the site fidelity. If I see them in the year round, so with or without a dome, potentially they are a coastal ecotype, and then the other one, if they come only during the season with the upwelling, they could potentially be. But it's also cautious what I say, but it's just a hypothesis.
And it's showing how many questions there still are. So as long as you can, maintain some funding, there's a lot of job security. We're always leading to another question. It's never going to be done. So I hope that you're able to continue. We had one last question about kind of the references to the time of year. So this was good timing. How much of the year are you in Nicaragua collecting data?
So when I started the project in 2016, I was focusing on the Northern Hemisphere whales. So I was collecting data from January to April, basically. And then when I discovered that there were whales from Antarctica coming, so the first one, the Northern Hemisphere, come from the US West Coast, and between January and April, and then they leave. And we have other whales that are humpback whales that are coming from Antarctica between, well, August, September, October. But I didn't know when they were coming, so I started investigating in July. So I did July, August, September, but in July you don't see humpback whales. So, but you do see dolphins, so that's valuable data. So I have, I don't have year-round data. So unfortunately, I don't have May and June, and then October, November, December. But still, I think I have enough data
A lot.
to assess if some dolphins are present year-round or not, or most
Yeah.
of the year, at least. Yeah.
Fascinating. Well, we wish you the best of luck with your research, with your degree, with your organization, with your education work. And we thank you so very much for doing this with us. And we hope that everyone has enjoyed this talk, and we will say goodbye.
Okay, thank you so much for an invitation. I really enjoyed it to give this presentation today, and maybe we meet each other in the Bahamas or in Nicaragua.
I
You're
look
welcome.
forward to it.
All
Bye.
right. Bye, Kel.